Hello everybody. This is an anatomical comparison between frogs and rats. And joining us today, we have Drew Lee, Harjit Sidhu, Sulan Jackson, and me, Nathan Kwa. First, we're going to start with animal backgrounds. And the rana foreri, commonly known as forest grass frog, is a frog that is widespread across Central American seasonal tropical forests. This includes parts of Mexico, Honduras, Costa Rica, El Salvador, Nicaragua, and Guatemala. These habitats tend to have rainy wet seasons and cool dry seasons. It belongs to the Animalia kingdom, the Chordata phylum, Amphibia class, Anura order, Venidae family, Lithobates genus, and L. foreri species. This frog can be found occupying plots of intentionally flooded agricultural land and various water transport systems, including sewers. It prefers to live in relatively moist habitats with a lot of water nearby because its porous skin absorbs nutrients from the water. Females will also lay eggs in water to be fertilized by males. As both the male and the female are in the water, the male will wrap its forelegs around the back abdomen of the female. The female releases the eggs, and the male simultaneously externally fertilizes them. This mating behavior is known as amplexus. The grass frog's diet typically consists of insects and spiders. It is predated upon by certain birds and snakes. Next, we have the rat, or the Rattus norvegicus. The Rattus norvegicus, or the common brown rat, is found in every continent except Antarctica, despite supposedly originating from northern China. They are particularly prominent in both Europe and North America, with urbanized and populated areas being where they primarily reside. As a result, they are commonly affected by abiotic factors, including human structures and changes to the environment. When not living in the presence of humans, brown rats prefer to live in damp environments, such as wetlands and riverbanks. The brown rat belongs to the Animalia kingdom, the Chordata phylum, the Mammalia class, the Rodentia order, the Muridae family, the Rattus genus, and the Norvegicus species. By living alongside urbanization and rapid expansion of humans, Rattus norvegicus lives amongst humans, using the infrastructure and sewers as a means of traveling between locations. Furthermore, the continuous food source in cities and urbanized areas allows these rats to thrive. The mating behaviors of brown rats can be classified as polyandrous, polygynandrous, or multiple of both female and male. As a result of being able to ejaculate multiple times, the likelihood of pregnancy is increased along with a decreased likelihood of stillborns and failed pregnancies. Brown rats, being omnivores, will eat almost anything. However, a large portion of their diet still consists of cereals such as grains, nuts, and fruits. The brown rat is predated on by many things that may coexist within their urban environment, such as cats, dogs, snakes, birds, and even weasels.
Okay. For the digestive system of a frog, the food starts from the mouth to the pharynx, esophagus, stomach, small intestine, large intestine, and finally the cloaca. For the passage of the digestive system of the rat, the food goes from the mouth to the pharynx, esophagus, stomach, small intestine, large intestine, and finally goes out. Beginning with the mouth's salivary glands, frogs have a unique salivary gland where the glands are actually not spread out around the mouth, but are located on the frog's tongue. There occurs the secretion of saliva for the purpose of having adhesive properties to catch prey. As for the rat, the salivary glands are located near the undersurface of the skin as well as inside the neck, which is basically opposite from the frog. However, the purpose of the salivary glands is similar, as a rat's glands also secrete mucus and saliva. These results of these differences can be found with the function of the esophagus. Since a frog does not have salivary glands around its mouth and neck, it has to go through a process called peristalsis, where the muscles contract in order to push the food past. For the rats, it may be much easier for the food to pass by the esophagus, which the mucus secretion aiding the process. The stomachs are very similar for rats and frogs. They both serve as sites for adding enzymes and breaking down the food in order for the food to be completely digested. For the intestines, except for the comparison of sizes between the two, with the rat's intestines being much larger, they serve as higher digestion organs. Moving on to the liver, a frog's liver is located when laying down on its back on the top right side. For a mouth, the liver is located at the bottom left. To add on, the liver of the frog has a three-lobed liver. The names of these lobes are called the left anterior lobe, the right lobe, and the left posterior lobe. The function of all these lobes is to continue the digestion process while also storing urine. Just like the frog, the rat's liver also has a few lobes. However, instead of having 3 lobes like the frog, it has 4 different lobes. To put it simply, it has a left lobe, a middle lobe, a right lobe, and a caudate lobe. The function of the rat's liver has many functions. Not only does it turn waste into more digestible foods, it produces bile, which also breaks down fats. Rats, on the other hand, do not have a gallbladder, whereas frogs do, as they need it to store bile. For the pancreas, rats use it to produce digestive enzymes, where frogs use it to secrete biles. The sphincter muscles for both rats and frogs are similar, as they both use it to push out digestive system products.
In the excretory system, the process of excretion is getting rid of waste. Therefore, the excretory system performs the function of excretion. In rats, the organs involved are the kidneys, ureters, and the urethra. In the rats, the kidneys are in the back of the abdominal cavity on both sides of the spine. They filter blood and secrete impurities, forming urine. The blood comes from the renal vessels, and the vessels are connected to the aorta and the vena cava. The ureters connect to the kidneys and lead to the bladder. The ureters are the tubes that move the urine from the kidney to the bladder, and the bladder is a single structure that stores urine before excretion, which is leaving the body. In males and females, the urethra are in different locations. In frogs, the main component of the excretory system are kidneys, a pair of ureters, a urinary bladder, and a cloaca. The kidneys have the same function in both frogs and rats. In frogs, the kidneys are more elongated and flat, located above the coelom. The ureter stores sperm and urine in male frogs and just urine in female frogs. The urinary bladder is a large structure that has thin walls. There are multiple tissue layers inside of the bladder. The bladder is also connected to the cloaca, and the cloaca is a sac that is connected to the urogenital openings, the anus, and the bladder. The cloaca is used to excrete waste. In female and male frogs, the excretory structures differ, as you can see in the pictures. And then for the reproductive system, the reproductive system produces gametes and supplies an environment for embryos to develop. The gametes are sperm and eggs. So in the male, it would be sperm, and in the female, eggs. The reproductive system in both females and males are different, as you can see in the picture. In male rats, the purpose of the reproductive system is to produce gametes and to transfer gametes, while in the female rats, the purpose of the reproductive system is gamete production, internal fertilization, internal incubation, and the birth process. The main components of the male rat reproductive system are the testis, epididymis, tubular vas deferens, urethra, seminal vesicles, and prostate gland. The epididymis collects sperm cells and stores them. Tubular vas deferens are what transport the sperm from epididymis to the urethra, which then transports the sperm out of the body. The seminal vesicles are on the left and right of the bladder, while the prostate gland is under the bladder. Both the seminal vesicles and prostate gland help to create semen. The main structure in the female rat reproductive system is the vagina, a pair of uterine horns, a duplex uterus, ovaries, and oviduct. Vagina divides into 2 uterine horns. The duplex uterus is very important because it will be able to hold multiple embryos, which will help form a litter. Ovaries are at the end of the uterine horns, and oviducts connect to the ovaries. In frog, the components of the male reproductive system are the vasa efferentia, testis, and urogenital ducts. The testis store nutrients that are essential for developing spermatozoa. Vasa efferentia are tubes that help to transport sperm to the uterus in the kidney. The uterus are also called the urogenital ducts. So those are interchangeable names. The key part of the reproductive system in female frogs are ovaries and oviducts. The ovaries produce the eggs, and the oviducts carry the eggs from the ovaries to the cloaca.
For frogs, their nostrils can be called these things called nares. The nares come in 2 different ways, which are the external nares and the internal nares. External nares allow the frog to breathe when the head is not completely submerged in water. And the internal nails are above the frog's mouth and serve as openings for the external nails. Frogs can also breathe through diffusion when underwater. Rats, on the other hand, cannot breathe underwater, so they have a nose instead. Moving on to the pharynx, a frog's pharynx is located behind the tongue and serves as a passageway for air to enter the lungs. For the rats, the pharynx is directly behind the nasal cavity, which also acts as a passageway for air. Frogs also have a larynx that acts as a voice box, but is much more complex than the larynx of a rat. Frogs do not have a trachea, whereas rats do. Both frogs and rats have a glottis or an epiglottis. For frogs and rats both, the function is to regulate what goes and what does not go into the lungs. Moving on, rats have a monopodial branching system for the lungs, which include the bronchi that reaches the lungs. The same is for frogs, except they do not have the same branching system. For the alveoli, or alveoli, frogs in their lungs have very small vessels that perform gas exchange, whereas rats have lungs that have alveoli in a specific pattern to help with oxygen absorption. Rats have a diaphragm, whereas frogs do not have a diaphragm to aid with diffusion.
As a result of one being a mammal and the other being an amphibian, rats and frogs have a few key differences and similarities in regards to how their circulatory systems function. Both the rat and the frog have a closed or cardiovascular circulatory system, meaning that the blood flows through the entirety of the cardiovascular system without leaving. Furthermore, both rats and frogs have two atria that pump blood into the ventricles, along with two exits out of the heart, each leading to either the body or the lungs. One very noticeable difference between these two is the fact that a frog has a heart with 3 chambers, while a rat has a heart with 4 chambers. Despite both having two atria, the frog only has one ventricle, while the rat has 2 distinct ventricle chambers, each carrying either oxygenated or deoxygenated blood. Frog's single ventricle has both oxygen-rich and oxygen-poor blood at all times. It is able to keep them separated through a system of complex arrangements. In order to transport oxygen to the rest of the body, the frog's pulmonary circuit first receives oxygenated blood from the lungs, while the pulmocutaneous circuit also receives oxygenated blood from the skin as a result of the respiration that takes place there. The skin of a frog contains a very large amount of vessels, allowing for gas exchange to occur. The right atrium then receives all the deoxygenated blood from various parts of the body. Both of these atria then pump the blood into the ventricle, which manages to send the blood into the correct corresponding places, despite both oxygenated and deoxygenated blood being mixed. Oxygenated blood is sent through the carotid arteries, which then sends blood to the head. Deoxygenated blood is sent through the pulmocutaneous arteries, which lead to the skin and lungs. A rat, being a mammal, has 2 atria and 2 ventricles in its heart. Deoxygenated blood from throughout the body enters into the right atrium of the heart through both the inferior and superior vena cava. This blood then flows to the right ventricle, which pumps out the blood into the pulmonary trunk to become oxygenated at the lungs. After the pulmonary circulation takes place, the blood returns to the left atrium and ventricle, which then pumps the blood back to the head and body through the aorta. After leaving through the aorta, the blood is divided into 2 sections, flowing through both the right and left common carotid artery and the right and left subclavian artery. The carotid arteries carry this oxygenated blood to the head and brain, while the subclavian arteries carry the blood to the front leg. A clear distinction between frogs and rats is the fact that the frogs are ectothermic, meaning their internal body and blood temperature is dependent on their surroundings. Rats, being mammals, are always warm-blooded.
So both frogs and rats have relatively simple brains in comparison to humans. Both animals have a more elongated brain to fit the body, with the frog holding a more elongated brain than the rat. In addition, both brains are bilaterally symmetrical. There is also a spinal cord present along with a set of nerves branching out throughout the body. The presence of the spinal cord also allows for quick reflexes in which the neuronal signals are not sent all the way to the brain, but to the spinal cord itself and back. Both frogs and rats have a spinal cord that is located on the dorsal side of their body, containing exactly 10 pairs and 12 pairs of cranial nerves, respectively. Frogs have 10 spinal nerves, and rats have approximately 34 spinal nerves. Furthermore, frogs have a specific reflexive reaction called the rostral wiping reflex, which is a form of elicitation to chemical stimuli. Rats also have a reflexive reaction that is very different from frogs. In an experiment done in 1962, shock stimuli caused rats to start a reaction called reflexive fighting. As for hearing, both animals contain ears that operate using the vibration of the eardrum to stimulate nerves via hair cells in order for the brain to interpret sound. The difference is that frogs do not have external ears. Whereas rats' eardrums are inside the external ears, both frogs' eardrums are visible on the outside of their heads. Both frogs and rats can walk and jump, but frogs are excellent jumpers relative to other animals. This is because they have dense and elastic tendons which can act like springs. So both being chordates, rats and frogs have a bunch of similarities in their internal and external structures. The most obvious ones are the relative locations of the forelimbs and head, and both animals have a long digestive tract, which begins at the mouth and ends at the anus. The relative locations of these features are consistent in both species as well, with the mouth obviously being on the head and the anus being between the hind legs. Both have a skull, which all members of Craniata have, and they also have backbones and jaws, which are representative of vertebrata and anastomata. And their skeleton is not made of cartilage, but it is calcified. The limbs are affixed to the skeleton by a single bone, and there is an opening that connects the nasal cavities to the throats. Internally, frogs and rats have very similar anatomies. They've got both a stomach with large and small intestines, and also a pancreas and a liver, and both also circulate blood using a heart, complete with a spleen and a system of blood transport. They breathe using lungs, and they've got brains and nerves, and both excrete urine and solid waste. Males of both species produce sperm in the testes, and females produce eggs in the ovaries. As for the general differences, frogs can also breathe through their skin as opposed to just using their lungs, and they will also have gills during their tadpole phase. In contrast, rats have tails, and the males will fertilize the eggs internally. Male frogs do not have a penis, and most frogs fertilize the eggs externally. Female frogs will therefore lay eggs before they are fertilized, whereas female rats will develop their embryos within the uterus in an amniotic sac. Looking at the phylogenetic tree shown here, the rat is placed in the Mammalia class, and the frog is placed in the Amphibia class. And to figure out the most recent common ancestor, one would trace from both of the clades in question the path up to the root of the tree, and you would pause where both of those paths meet. In this case, this would be at the node separating amphibians from amniotes. Therefore, the most recent common ancestor between frogs and rats had to have been a tetrapod lacking an amniotic sac for its embryonic development. And it must also have lived in an aquatic environment, considering that tetrapods evolved from lobe-finned fish, and that living in aquatic environments is a relatively basal trait that frogs possess. This makes sense because more terrestrial tetrapods would have to create an internal aquatic environment far away from sources of water in which to lay eggs. This is what probably led to the evolution of the amniotic sac in the context of unoccupied niches of non-aquatic terrestrial environments. And that is all from us here today. Thank you for watching, and we hope you learned something new.